Microorganisms are organisms that cannot be seen with bare eyes.
We observe that microorganisms is around us, like for instance in the doorknob, in your laptop, in your mouse, and we conducted some experiment that makes and shows that microorganisms are around us. Microorganisms contain a morphology of smooth, some are wrinkled, some are contoured, some are concentric.
In order to see these microorganisms, we need to use microscopes to magnify the microbial cells.
In order to see the specimen very clearly, we have to put the immersion on to shorten the wavelength and reduce the reflective index.
We found a microorganism under microscopic lenses. It is called ciliated infusoria. It's a kind of zooplankton.
We have to prepare, smear and do simple staining before we put the specimen under microscope.
Fixing the nature's bacterial enzymes also enhance the adherence of bacterial cells to the microscope light. We use methylene blue and crystal violet for different purposes. Methylene blue is used in simple staining, whereas crystal violet is used as the primary stain in gram staining.
When you are dealing with staining, time is a very crucial point because when you give it much time, it becomes overstained.
There are differences between agar media and nutrient broth. Agar media are prepared using nutrient agar powder, whereas nutrient broth media are prepared from tritone soy broth powder. Three physical forms of culture media are liquid media, semi-solid media, and solid media. Solid and semi-solid media contain a solidifying agent, whereas a liquid medium does not. In the strip plate method, bacterial mixture is transferred to the eggs on agar plate using inoculating loop and then streaked out over the surface. The key principle is by streaking, a dilution gradient is established on the surface of the agar plate.
The aseptic technique is very important to prevent contamination of culture from foreign bacteria inherent in the environment.
Turbidity of cell suspension is measured to estimate the number of microorganisms present in the sample. Spectrophotometer is used to measure the turbidity of cell suspension. There are four phases in bacterial growth, which is log phase, lag phase, stationary phase, and death phase.
However, we do not get the four different phases. We get up to lag phase and just to start log phase due to slow adaptation of culture, environment factor, and also not optimal temperature in the laboratory.